Good evening, ladies and germs. Welcome to the opening night of the 2018 Wisconsin Science Festival. My name is Laura Heisler. I'm the founder and director of the festival, and delighted to welcome you tonight. I want to start out by thanking the many sponsors who make the festival possible. You can see them all here, and in particular, I'd like to thank Thermo Fisher Scientific, who is the presenting sponsor for this evening's event. I also want to take this moment to shamelessly plug a few upcoming highlights that are taking place over the next 24 hours that you're not going to want to miss. So at this time tomorrow night, we will all be downtown colonizing the Capitol Square with science. So if you ordinarily enjoy going down to the Capitol Square to have a beer, grab some dinner, go to a shop, you're going to want to do that tomorrow night because more than 24 different vendors all around the square and up and down State Street are featuring science as part of what they ordinarily do. So check it out. You can find all the details at wisconsinsciencefest.com. And then tomorrow, a little bit more than 24 hours, I guess I'll say. Saturday at noon, we have a very rare opportunity to hear from Frances Halzen, who was one of the lead scientists on the recently much talked about discovery of a new kind of neutrino through the Ice Cube project in Antarctica that you may have heard about. It's a global research study. UW-Madison is one of the lead participants in that study, and Frances Halzen will be talking about that here in this room at noon on Saturday. And then Saturday evening, right back here, a very unusual free episode of Gastropod Live, which is a phenomenal podcast all about the science and history of food. Yes, there will be things to eat. It will be very cool. You want to be here for that. You don't want to miss it. So with that, can I see a show of hands of folks who have been to a Big Ideas for Busy People before? Excellent. So you know that this is not your ordinary science talk. Can we get the timer up to show us what the timer looks like? So we are hearing from some of the best scientists at UW-Madison and beyond. And ordinarily when you hear from them, they talk for, a while. Tonight they get 5 minutes. And if they go over 5 minutes, we'll have that gong maybe a little bit softer next time. And then they have to stop. And then we reset the timer and they get another 5 minutes. You can game the system a little bit. Any ideas of how you guys might game the system for them if they get gonged? Just shout it out. What's a question you can ask them to game the system? What were you going to say before you got gonged? So if you really want to know, you can do that. And if you don't want to know, don't ask that. To do the Q&A, to move it along quickly, we're going to use this. This is a microphone. It's actually embedded in this nice soft foam thing. It will not hurt you. So we're going to have you guys toss it between each other. You see someone who has their hand up. Doesn't matter if they were the next one up. Just toss them the mic. Okay, Patrick, here it comes. Like that. Okay, so don't be afraid. And we'll just keep it moving along like that. In addition to the unusual features of this kind of science talk, tonight we're trying something different for Big Ideas for Busy People. It's going to be a 4D experience and full-on smell-o-vision. So you will all be participating in this evening's event. And we're going to start that right now by handing out our wonderful, lovely assistants are handing out envelopes. You're all going to get an envelope. Please do not open the envelope now. Okay, you're going to get one. If you don't get one, let us know that you didn't get one and hang on to it until I tell you to open it, which I'm not going to do now. Okay, so now what I would like to do is hold myself accountable to the same standard that we're holding our wonderful scientists accountable. So I'm going to ask you, Wes, to go ahead and start the clock. And I'm going to take much less than 5 minutes to introduce all the speakers now, because then they're all going to come up and we're not going to introduce them again. They're just going to go one after the other, okay? So our first speaker, she is an assistant scientist in the UW-Madison School of Pharmacy and also the interim director of that school's global health program. She is Laurel Lagenza. Please, Laurel, stand up and wave. There she is, Laurel Lagenza. Okay. Second, all the way from UC San Diego, a visiting scholar as part of the Wisconsin Book Festival. He is an anthropologist, performer, author, director, actor, and instructor, Patrick Anderson. A little closer to home, like a few feet above us, is the director of the Wisconsin Institute for Discovery and professor in the Department of Plant Pathology, whose research focuses on microbial diversity with a particular emphasis on microbes found in soil. Please welcome Dr. Jo Handelsman. Wave, wave, there she is. And across the building from Joe, also in this building, is a very prominent virology researcher with wide-ranging interests and is the director of the John and Jean Rose Center for Research in Virology at the Morgridge Institute for Research, also in this building. Please welcome Professor Paul Ahlquist. And then finally, to bring it all home, we will hear from Dr. James Conway, who's the Associate Director for Health Sciences at UW-Madison Global Health Institute, and is also a pediatric infectious disease specialist who works domestically and globally with programs designed to improve the lives of children. Jim Conway. All right. How'd I do? All right, I got plenty of time. Okay, so what I'm going to do now, because this is going to move quickly, you guys, I'm going to pull up all of the slides. They're all in one massive slide deck. And we are going to reset that clock. And I'm going to welcome Laurel Lagenza to get us started.
Thank you, Laura, and I'm thrilled today to be sharing with you some research we've been working on at the School of Pharmacy. Consider this beautiful forest and the vast array of wildlife within it, as well as plant species and even bacteria. This forest could be anywhere in Wisconsin. Also consider that on your body, you also have a whole ecosystem of bacteria and fungi that are living on and within you. And that can be variable between populations, globally, nationally, and even across Wisconsin. So one way that healthcare providers think about the ecology of pathogens that might be causing an infection is a table called an antibiogram. And this is an example here. And as you may be squinching your eyes, it's very difficult to interpret and read, but I'll walk you through it, that what this is showing is just a list of pathogens, and then the percent probability that in this population, the antibiotic would work. So for example, if we had a number like 74, that means that 74% of the population would respond to that antibiotic. Or 1 in 4 patients, the bacteria that they have that's causing the infection would be resistant. So I looked at this table and I saw that it wasn't being used effectively in healthcare systems as much as it could be. And we wondered, was there another way to present this data so it was more intuitive, like a weather map? When you look at this weather map, you see immediately where the storm is. You know that if we are in Madison and this storm is headed our way, we need to get an umbrella, think about flooding, and be very concerned. So you have this immediate reaction. Similarly, you could list all of these cities with the probability of rain, and it would be a very similar numerical table like this. So my research group said, well, let's ask physicians how often they use this, and can we think of ways to make this more easy to interpret? So we started collecting these antibiogram tables, and we aggregated them across 3 different years. And then we interpolated the results between health systems. And what we found was that working with the state geography department, we were able to interpolate the results to create these vivid, beautiful maps where you can see where resistance is changing and also where there's higher and lower resistance patterns. Then we said, can we use this data to create an interactive tool where a healthcare provider could immediately see what the pattern is for an antibiotic and a pathogen? So in this prototype that we developed with the state cartography office and my collaborators at the School of Pharmacy, a provider would immediately be given an advisory that if the patient had been hospitalized or had similar infections, then they would be at higher risk of antibiotic resistance if they already got antibiotics. And then we consider what pathogen might be causing the infection. And we can also then see an immediate list of different antibiotics and the probability that they would treat that patient's infection for that specific patient and where they're from, considering their geographic location. And now this map here is now showing one antibiotic and very high susceptibilities across the state, which we expect. But interestingly, we're starting to see areas where the percent susceptibility is starting to drop below 90%, which is unexpected. And similarly, for each of these antibiotics, we see very different patterns. So my future research is now looking at, can we study what's going on in these different areas that might be contributing to higher and lower resistance patterns? And we can also then consider what other population factors might be related, such as, is this a rural area or is it urban? Is it an area close to farming where they might use antibiotics as well? What other factors can we think about in that region and that population? And what about in areas where we don't have strong resources available that's monitoring antibiotic resistance? And I hope that a tool like this can reach beyond the university across the state to provide this interactive, usable tool for healthcare providers, but also increase awareness with the population.
Great job, Laurel. That was great. Okay, so I've got the catch box. Who's got a question? All right, I'm going to try to throw. Alan's going to help me. All right, Alan, here we go. Excellent.
Is it working? Can you hear me? Excellent. Is this tool actually available to practitioners outside of UW at this point, or is it still just research?
At this point, it's in a prototype phase. So it's interactive and functional, but it requires a password that only my research team has right now. Our goal is that it'll be fully publicly available by July 1st, 2019. And we're currently recruiting health systems to give us a higher resolution data so we can create even more valid maps.
So what's going on in Oconto County?
So in Oconto County, so please note that this is beta test data and not yet published, but I can tell you exactly what's happening is we're seeing an emergence of different bacteria that are highly resistant. So what happens is sometimes bacteria have genetic properties that make them resistant to virtually all of the antibiotics we expect would work. And that creates an outlier in our data. And we're doing different things to most appropriately adjust for that.
Is anybody tracking antibiotic resistance in animals to coordinate it with people?
Yeah, so I've talked to some colleagues at the School of Veterinary Medicine, and there are some researchers here that are looking at resistance in some of those animal pathogens like salmonella. So I am interested in paralleling that. The trick is that the pathogens that cause human infection and animal infections, there's some overlap, but there also is a lot of differences. And there's also a lot of differences in what antibiotics you use to treat infection. So I'm certainly interested in exploring that relationship, but it's not going to be as easy as comparing cities where we have the same pathogens and the same antibiotic options. Yes? That I'm not familiar with, I'm sorry. Human cow, they probably are. So the School of Veterinary Medicine, I imagine, is looking at the different variations of salmonella like that. I'm not well versed in the types of those, thank you.
And if you have a question, and wait for the mic to come to you, thanks.
There's some lines on the map. Are those highway systems?
Depends which.
They're real faint lines all over the map. What I was wondering, could you find a way to highlight the county boundaries?
Yes.
That might be
so
more helpful.
Unfortunately, my video didn't play, but in another version, it's really easy working with geographers to click and add different layers. And one easy one to add is both cities and county boundaries. So I'm interested in looking at county boundaries, but also geographic boundaries, such as highways, riverways, watersheds, and how that might affect not only where people live, but where a transfer of water and pathogens happens.
In the front row?
Yeah, the video is really cool. I have a whole minute. Okay.
We're really sorry. It is very small.
It worked earlier. I'm really sorry. Other questions? Yeah.
Is there a seasonality to the maps?
Currently, right now, we have it categorized by year. So we have distinct data sets from 2009, 15, and 13. But certainly when we look at patterns of infection, that varies by month and with season. But not with the current data set. My future data set that will hopefully have that higher resolution of data, I could create maps that are by season. That's a great comment. Thank you.
Time for one more if you're fast.
Just a gentle suggestion that you consider plotting also on your antibiotic resistance map the localities of concentrated animal feeding operations.
We've thought of that as well. Thank you.
Thanks so much,
Thank
Laura.
you, everyone.
Great job.
Thank you.
Okay, all right. So now we go back to getting you guys involved. So take out that envelope. This is your first instruction. Take out that envelope, find your sticker, put it on. That's all you have to do right now. It's going to get more complicated as we go. All right. Next presenter, Patrick Anderson.
Hello, can you hear me? Hi, my name is Patrick, and I am not a scientist, but I do not like the sound of that Salmonella Heidelberg thing you were talking about. That's my confession. I was trained in anthropology and performance studies, and I'm now a professor of communication, ethnic studies, and critical gender studies at the University of California, San Diego. My research concerns the role of violence, mortality, and pain in the experience of subjectivity and personhood, so a little distinct from what we're doing here. I've written books about hunger striking, force feeding, the diagnostic history of hysteria, anorexia, and similar kinds of conditions, and most recently, what I'll be talking about in the longer talk after these short segments, my most recent book. This book is meant to document the experience of being a patient within the labyrinthine American medical system that I'm sure a lot of you have also had horrific experiences with. It's that book that's brought me here today. And it's intended to document the experience about 15 years ago of me falling first into a coma and then spending nearly a full year in several hospitals in San Francisco, where I was then finishing my doctorate at UC Berkeley. I'll be talking about that book in a few minutes. But first, I want to start in 1912, shortly after the sinking of the Titanic, when a group of Tony Londoners arranged a charity fundraiser at the Royal Opera House in Covent Gardens in London. The organizers contacted the British author Thomas Hardy, who some of you may be familiar with, and commissioned him to write a poem that would be read at the event. They hoped for a sentimental ode to the many lives lost in that disaster, but to their great disappointment and with widening controversy, Hardy wrote instead a poem that was a kind of love letter between that fated ship and the iceberg that brought it down. The poem is called The Convergence of the Twain. I love that title. And rather than mourn with droning romanticism the wealthy friends and relatives of those gathered at the opera house, Hardy instead indicted the spectacular events that had produced the Titanic in the first place. He also fantasized about how the ship and the berg were fated for one another. That was the love story. My favorite line, nearly 2/3 of the way through the poem, marks what Hardy called the intimate welding of their later history. I love that line, the intimate welding of their later history. It's that idea that inspired my book, Autobiography of a Disease, which narrates the story of my nearly fatal illness and long, slow convalescence from the perspective from the narrative perspective of the virulent strain of drug-resistant bacteria that had occasioned my collapse. The image you see on the screen, hopefully, there it is, a fluorescein angiograph of my scarred and infected retinal tissues, represents a documentation of the bodily trauma those bacteria caused. But this image is also, to me at least, an image of profound beauty. I grew curious about whether and how the bacteria that had produced it might have a sense of that beauty. I think they look so tender here. Or experience it in some distinctly non-human way, of course, differently than the medical professionals who so diligently strove to eradicate them. The intimate welding of their later history, the bacteria and I. I wondered if there might be another way to understand microbes, one that considered them not just as invasive enemies, but also quite simply as living beings. And it is this that I will be inviting you to consider with me in a reading and talk that follows the five of us. I'm a storyteller. We know how to time things.
Yeah, really. Great job. All right.
Go ahead and gong me, and let's move on.
We can wrap it. That's right. Excellent. So thanks so much, Patrick. Okay, I've lost track of where oh, it's right here. Here's the microphone. I have to lob it again. Okay. Questions?
I would also be happy to cede my question time until after the later talk, if that makes sense.
Any questions for Patrick? There's one way in the back. I cannot throw that far.
Sort of close.
What type of bacteria exactly did you have?
It was MRSA, community-acquired MRSA. And this was in an era when there were beginning to be news stories about young, otherwise healthy people suddenly collapsing in locker rooms and so on, and passing away very quickly out of the blue. I was fortunate, on one hand, not to be someone who died. I was one of the few in this particular moment, before a standard of care for this particular trend had been fully fleshed out, as it were. But I didn't die. And as I found out later, one of my many surgeons ended up testifying about my case before the Senate, strangely as a way of increasing reimbursements of doctors who take on difficult cases. Not what I would have chosen to talk to the Senate about, but it was something. This is all a long answer to your question. It was MRSA. So it was methicillin-resistant Staphylococcus aureus.
Other questions? Okay, well, Patrick is going to be doing a talk from his book in this room after we're done. We have other activities after we're done as well that I'll tell you about later. So with that, thank you very much, Patrick. Okay, and we will move on to Dr. Joe Handelsman. Oh, wait a second. Sorry, I think. Should you really be here, you guys?
Big ideas for Contagion?
Yeah. I'm a little bit worried that you guys might be contagious. Are you a little itchy?
Very good. Oh, itchy. What do you got? 104, 105? 103.8. You're almost there.
Yeah, okay. I'm a little worried. I don't know, Jim, if we should be a little bit worried. Jim thinks that's okay. Jim thinks we should be. No, he does. Jim doesn't think we should be worried at all. Yeah. Yeah, oh, okay, all right. Yeah, so I guess everything's fine, and we'll just, let these guys find a seat.
Hey, Stan, were you hanging out with that person that was sick, Jenny, on like 3 or 4 days ago? Was that her? Oh, I think she got sick.
All right, well, okay, so you guys gonna find your way to a seat? Okay, all right. Yeah. I don't know what could go wrong. All right, well. All right, while you guys are finding your way. Yeah, okay. Okay, well, I guess they're handing out some goodies for you. So do hang on to them. If you get a goodie bag, just keep it to yourself right now. Just keep it to yourself as best you can. Okay, we're going to let you guys find a seat. We're going to move it along. Okay, and before we do, I want to make sure, Joe, the podium doesn't come off the stage. So thank you. It's been migrating. Excellent. Okay, well, while these guys are migrating their way along, we will let Dr. Joe Handelsman take the floor.
Okay. Well, thanks. And I'm glad I was up here and not down there. Maybe I won't get sick. So we've heard about antibiotic resistance at the state level in Wisconsin, and we've heard about it at a very individual and personal level. And now I want to take you to the global level and talk about this growing crisis that is causing great concern in the medical community and beyond. It comes from two facts. First, there's growing resistance among some of the major pathogens. This is just one example. This is the Neisseria gonorrhea pathogen that causes gonorrhea. And this is over the first 8 years of this century. And you can see the rapid increase of resistance of gonorrhea strains to the major antibiotic Cipro that's used to treat them. This is just one of many, many examples. At the same time, and this is the really scary part, we've essentially come to a grinding halt in terms of discovering and licensing and commercializing new antibiotics. So in the early days of antibiotic discovery, and that was from the 1940s to the 1980s, we were seeing 10 to 20 new antibiotics released every year under FDA approval. But since the 80s, the pharmaceutical companies have pulled out of antibiotic discovery. We, as of a few months ago, were down to 3 companies, 3 of the major pharma companies. And then just as I was saying that one day, I found out that Novartis pulled out. So we're now down to 2 major pharma companies that are doing antibiotic discovery and commercialization. And so as a result, from the 80s until now, we've gone now down into the single digits. And we have typically 4 to 6 new antibiotics released per year. That's not nearly enough to address the problem that is growing across the world of antibiotic-resistant bacteria causing human infection. The WHO, or the World Health Organization, predicts that by 2050, antibiotic-resistant bacterial infections will be the leading cause of death internationally, the leading cause of death. So this is not a trivial problem, but we do have some warning before it becomes epidemic or pandemic proportions that this is going to be a problem. So in 2012, I was a professor at Yale University, and I started a new course with the hope that I could simultaneously excite undergraduates about science and begin to address this crisis. So one of the things that is important in the antibiotic history is that most of our antibiotics, about 75%, come from soil bacteria. And so it's a very relatively simple thing for students to collect a soil sample from somewhere near their classroom, or to cut a deal with a farmer or a gardener or somebody who has interesting soil and who can collect it for them, to culture bacteria from that soil sample, isolate individual strains that are growing on that original plate, test them for inhibition of major pathogens or safe relatives of those pathogens. So these clear zones that you see are indications that the pathogen on the plate is not able to grow because there's an antibiotic coming from the center, which is a piece of filter paper carrying the bacteria. So they can quickly screen their isolates for antibiotics, and then they can characterize the producing organism and the antibiotic. And in the longer run, we'd like to be able to get chemical structures of many of the antibiotics that are discovered by these students. Most of them do not get to this level in their classroom, so we're trying to develop a chemistry hub here at the Wisconsin Institute for Discovery that will do the chemical structural analysis of many of these students' isolates and the chemicals that they produce. The number of institutions has risen exponentially. So in 2012, Yale was the only place teaching it. I had a total of 6 students the first time I taught it, not a popular course. Today, we teach 10,000 students across the world. And you can see this increase in the number of institutions. We're up to about 300 as of this year. We train instructors, which is one of the reasons that it's a very popular course. We teach them how to teach the course, and then they go back and teach it to generally great acclaim. So the goal is, if the antibiotic industry is going to disappear, we will replace it with students. And we've taken the concept of crowdsourcing, which has been a very successful way of discovery, and turned it into student sourcing.
Brilliantly timed. Okay, questions for Dr. Handelsman?
Do you have plans, or have you already tried to make the crowdsourcing more publicly available? So, for example, anybody can dig dirt, and maybe even having pre-made petri dishes or chemistry kits, we can do at home.
So we're very eager to get soil samples very broadly, and that's something that we're starting to do in the state of Wisconsin, just as an experiment, and then we'll send that out to other sites as well if it works. The only problem we have with giving the Petri plates and culture conditions to everybody is that it is possible to culture pathogens, human pathogens, from the soil. And so the American Society for Microbiology and the NIH have regulations about how we're supposed to culture from soil and when we do it with students, and we can't really meet their criteria if citizens are doing it on their own. So we have people come in to WID or the Discovery Building to culture under controlled conditions, but we're a little bit afraid to turn the plates out to the public.
Hi, do you have any molecules that are getting to any human trials yet?
No, we have several new molecules. So we've shown that we, in fact, can discover new compounds using this process. But it's a long road to typically about 10 years, actually, to get a drug to market. And that's because all sorts of testing has to be done to make it as effective as it can be. We often call it making it a druggable compound. So changing its chemistry so that it has the right properties to get taken up into the blood and distributed around the body. Every compound has to go through safety trials, and so we haven't gotten to any of that yet, mainly because we don't have funding. The students do all the first part of the project, discovering the antibiotic-producing organisms, and that doesn't cost us money because the institutions support the course. But unfortunately, we don't have money to take it the rest of the way, and we're working on that really, really hard.
Joe? What's the latest advancements in culturing previously unculturable soil bacteria? I think it's a very minor percent that will pop up in this kind of technique. Has there been advancements there?
Yeah, so that's an interesting point. About 1% of the bacteria in soil are readily culturable on culture media, and that is a limitation to discovery. But what we found is that there are still a lot of antibiotics to be discovered from the cultured organisms. And the antibiotic industry kind of put out the word that the soil was tapped out. That was the phrase that was used in the 80s. And it's simply not true. We can't find data to support that assertion, and our data certainly argue against it. Yes, there are a few tools that have been developed for culturing bacteria from soil that previously weren't culturable. That generally has to do with either putting them under starvation conditions or giving them their friends and buddies to play with so that they grow more effectively. Many of them are dependent upon other organisms in the soil. If you co-culture them, it's easier to coax some of the soil bacteria to grow.
In the back?
Given that there will be a growing market for their products, why are the pharmaceutical companies not investing in antibiotics?
Is simple time that the antibiotics have to be taken. So antibiotics are typically taken for 1, 5, or maybe 10 days, unless you're extremely ill, like we just heard about, then it becomes a much longer proposition. What they discovered in the 80s was drugs like Lipitor or asthma medication, anything that people will take for the rest of their lives, is just much more lucrative. And so for the same registration costs, they can generate a lot more profit. Now, I agree with you that in the longer term, there will be a very lucrative market there because people will be dying of infections all over. And now they're certainly dying, but not as frequently. The problem with that is that most of the pharmaceutical companies are working on a very short time frame in terms of return on their investment. And so it's very hard to say that they're working now on antibiotics that will be needed 25 years from now.
Thank you so much. That was fantastic, Joe. Now, thank you. Excellent. So we're going to take a moment and take a look at those bags that some of you got. So if you are holding a bag, those of you holding bags, will you stand up? See your bags, wave them high. Okay. Now take a look at the sticker that you're wearing. If you have a green sticker, please just drop the bag on the floor and you can sit down. Okay. If you have a red sticker, go ahead and open up the bag and take out one of the objects. Just hold it up for everybody to see. It's measles. It's measles. You have measles. Congratulations. You are infected with measles. Now, I'm going to give you guys some instructions, and everybody's going to get involved. But please don't do anything until we restart the timer, okay? So what you're going to do when I give you the signal, which I'm not doing, is you're going to take handfuls of the measles out of your bag, and you're going to pass them to the people next to you. People that get handfuls, if you have a green sticker, just drop them on the floor. We'll get them later, don't worry. If you have a red sticker, pass some more along. Anybody who's passing with a red sticker, keep one for yourself. So make sure if you have a red sticker that at the end of all this, you're holding one measle. But you're going to pass handfuls, 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 okay? And we'll gong when it's done time. Does everybody understand the instructions? You ready to start passing? Okay, let's start the clock. Go. Pass, pass, pass. Pass along. If you've got a green sticker, you don't pass, you drop, you don't pass. If you're wearing green, you do not pass. If you're wearing red, you pass. If you're wearing green, you do not pass. If you're wearing green, you do not pass. Drop them. If you're wearing green, drop the bag. If you're wearing green, do not pass the bag. I see people wearing green passing the bag. People wearing green, stop passing the bag if you're wearing green. Okay, green people, stop passing the bag. Stop passing the bag. Drop the bag. All right, excellent. Okay, that was complicated. It's a good thing that the viruses are smarter than we are, which they are. Okay, so great. We will revisit this momentarily, but now we are going to move on to our fourth speaker, Professor Paul Ahlquist.
Well, so here we go. So I want to continue this transition to viruses, and I'm going to talk to you about the need for broad-spectrum antivirals. So as the news keeps reminding us, humans are continually threatened by many dangerous pathogens like Ebola and Zika and HIV and many others. Now, all of these viruses crossed over into humans from animals, and this crossover trend of emerging dangerous pathogens into humans is not only continuing, but is accelerating because humanity is constantly exposed to an essentially infinite reservoir of serologically distinct animal viruses that are continually trying to figure out how to circulate in the human population. Now, against this unlimited, increasing pool of dangerous viruses, we have a very limited ability to generate new antiviral drugs and vaccines, continuing the sort of idea that Joe was generating. To deliver a new drug or vaccine to the clinic typically costs on the order of $1 to $2 billion with a B. So we can do this very rarely, and we have lack controls for almost all viruses currently. So to make progress, we need to change this game. And for the drugs that we do develop, we need to generate drugs that target not only a single virus, like the agents we have now, but target many viruses in one step. One attractive way to do this is to target drugs not against an individual virus, but against host genes or pathways that are used by many viruses. So the job of a virus is to infect a cell, to replicate through complex pathways, and to release thousands of infectious progeny virions to continue the infection, like the bags you're passing around here. It turns out that to do this, a typical virus has 8 to 10 genes, which is not enough to control these processes. So the way this works is that the cell has about 20,000 genes. And viruses are intrinsically dependent, all viruses, on hijacking these host genes and pathways and redirecting them to direct their replication steps. Now, work in our lab and other labs shows that many viruses require common host pathways. So if we can modulate or perturb these well-selected common host pathways, we can block many viruses in a single step. So examples of host factors and pathways that are used by many viruses include those that are involved in the synthesis, modification, and reshaping of membranes, which are important in many different infection processes. Pathways involved in the folding and assembly and recycling of proteins, which carry out most functions in the cell, and many different transport processes that move proteins and molecules around in the cell. The point is that viruses depend on a great many different cellular pathways, and we can choose among these and perturb them singly or in combination to generate strong, broad-spectrum antiviral effects. Now, some of you are thinking that we certainly can't afford to perturb host cell processes because that would make people sick. But in fact, there are many results that show that this is not a disqualifying issue. So again, we often find that host genes that are required by viruses, we can completely remove them from the cell without making the cell sick. It's often true also that we find that it's sufficient to just tweak a pathway rather than to completely knock it out. Another important point is that other than antimicrobials, all of the drugs that we have, all of the drugs that you're taking, are against your own host cell proteins and pathways. This includes many drugs against chronic disorders like diabetes, high cholesterol, hypertension, arthritis, and so forth, where, as Joe was pointing out, you have people taking these drugs every day for the rest of their lives. Against that, we only need to treat for a day or perhaps a couple of weeks. So I'll just remind you of the take-home lesson and take any questions.
Yeah. Right. Questions for Professor Alquist in the back? All right.
How much does farming, specifically animal farming, have an effect on the mutation and development of new viruses? Like, if we got rid of, or if farming practices changed, how much could we slow the development of new viruses?
So, as you're implying, the interface between humans and animals is an important point for transferring viruses and for the development of new diseases. A prominent example of that is the transfer of avian influenza viruses between farm operations and people, which has happened in most continents in the last few decades. What's getting more attention now, actually, than farming is the expanding encroachment and wild environments. Obviously, we've been living with most farm animals for a long time, so a lot of people think we've sort of reached an equilibrium with those interactions, but we're now interacting with many different kinds of species that we have not before as we move into rainforest and other kinds of environments.
Sorry. So we know that viruses evolve and do so rapidly, but we also know that our own DNA can change. How has human evolution been factored into the data that predicts the future of antiviral drugs and their efficacy?
So I should point out just at the outset that as you began to indicate, the viruses mutate at a ferocious rate that is tens of thousands of times faster than any changes in humans. So we're talking about completely different scales. At the same time, virus infections have had major effects on humans. You can see this in a variety of effects, including remnants of viruses in our own chromosomes from past epidemics. There is also evidence of what's called purifying selection in populations, which is a discrete way of saying that much of the population died off and made a major change in certain gene variants in the population. Got another question over here.
Okay, heads up.
Hi. So you mentioned temporary treatment is sufficient for viruses, but if you're doing gene knockout, that's not going to be temporary, because especially if it's a whole body treatment, it's going to change the functioning of those cells indefinitely, right?
So we use genetic methods in the laboratory to identify genes that are involved in these processes, and currently that's much more powerful than chemically derived methods. But of course the current medical model is to identify a target genetically and then develop a small molecule chemical to perturb that target. So that will assuredly be the major way that these sorts of approaches are played out in the short term. In the longer term, I would be hopeful that we'll develop further genetic methods that are not threatening to people, that can be used to accomplish these things much faster and without the tremendous investment of time and effort and money that's required to get a chemical to duplicate the genetic results from the lab.
Seconds for another question?
Thank you.
How did the pandemic end in the 1918 or so? And is there anything that we can learn from it, because it just sort of ended?
Right, so you're referring to the 1918 influenza epidemic, one of the major events in public health history. So basically that virus moved across the entire world in a matter of months, and reaching even small islands in the Pacific in a time when people were traveling by steamship. Basically, it infected everybody, and you either died or you became resistant. So the next time it moved through, there was protection resulting from that, which may relate to things that Laura is going to have to share with you.
Yeah, thank you so much. Thank you, Paul. Great. And on that note of infections moving through, we're going to bring our activity to its climax. So if you would all please stand. Thank you. Okay, now if you are holding a measle, wave it up high, hold it high. Okay, you are sick. I'm really sorry. You need to sit down. You saw what those guys were like. So you are sick. You probably have a fever 104. Take a seat. You need it. Okay. Now, you'll see that one half of the room had a lot more people sitting than the other half. So I'm guessing some of you have figured out what the significance is of the colored stickers. Shout it out. Yes, all right. Now, okay, so anybody have a red sticker not holding a measle? Yeah. Okay, so you are protected by the herd, and we are going to hear much more about herd immunity from Jen Conway. Congratulations. Now, everyone else can sit down. Great job, everybody, and most of you are well. Those of you that aren't, take the measles with you, share them with your friends. I do want to thank Wes Marner for devising this activity, Janine Ennis for contributing to its execution, and of course our measles-infected actors. They're really fine. Travis Tangent and Dan Murphy, they're fine. They're going to be back at the festival tomorrow. Let's give them a hand. Thanks so much, you guys. Great job. Okay, now we're going to give Jim a few extra seconds on the clock. I apologize to other presenters, but Jim spent a lot of time working with us on the game, and he earned himself another 30 seconds. So if you ever want more time, you can just help us with our games. Okay, so go ahead, Jim. Thanks so much.
Thanks very much, and thanks for having me. I have to tell you one thing that my disclaimer is that I'm terrified and horrified that I ended up with a red sticker. So I am admittedly a vaccine zealot. And have had pretty much every vaccine known to man, so the odds that I would actually be the unvaccinated person would be almost zero. Now, I'm also here to be a little bit more on the encouraging side because I'm relatively terrified by what my preceding speakers were talking about, and so I'm going to give you some hope that we actually have some other ways to manage these situations and perhaps prevent some of the issues that these folks are talking about as far as treatment. So this is the good old days that we used to live through, and although some people are fond of the good old days, I'm perfectly happy that we don't see chickenpox and invasive group A strep necrotizing fasciitis, that we don't see polio, we don't see kids with measles very often, except for this half of the room, we don't see kids with mumps, we don't see kids with congenital rubella, and we don't see kids with Haemophilus influenza invasive disease very often. I am happy that we have been able to eradicate, or at least control up until now, most of these conditions. And how have we been able to do that so successfully in the United States? Through vaccines. Vaccines have, without question, been the single biggest thing that we have ever done as a society to prevent disease. And so at the end of the last millennium, when the CDC was hunkered down in its bunker, waiting for all the airplanes to fall out of the sky and the computers to melt, adding up all of their successes over the previous century, vaccination comes out by far number one for prevention of mortality and morbidity. So why do I then need to talk about our state bird, the cow? Well, because the herd is actually what really protects most of us. So this is what we've been living through and what you've been dealing with so far. We got a little bit different on the colors here, but in most of society as we function today, the yellow people are the immunized people, but there are people that either cannot be immunized because they have some underlying medical condition or an allergy to the vaccine or perhaps a religious opposition or may just not have access to vaccines. A person with one of these diseases walks in, and fortunately there's a firewall around them thanks to all the other people that have been vaccinated that protects these other vulnerable individuals. This is also unfortunately what allows people who choose not to vaccinate to get away with it because of the goodwill of all the other folks. Now we have some critical numbers that we're looking for from a public health standpoint, and this is what I mostly work on in my role here in the state of Wisconsin as well as globally, is trying to improve herd immunity by improving either access or system delivery things around the world. We have some critical numbers that we're always looking for in trying to achieve the herd immunity that we want. And although these numbers seem very close together, they actually are about 100% apart because 80% of the people immunized means that 1 in 5 people are susceptible to the disease. But if we get over 90% of the herd immunized, that's only 1 in 10 people that are susceptible. And you can certainly see then how much less likely it is that you're sitting next to one of those susceptible, vulnerable people that may then transmit the disease to you. These are actually based on real scientific transmission models, but the issue here then is that we're really living in a close and narrow space when we're working on trying to improve immunizations. Now this is a little bit of a draconian look at these things. This was an editorial from the New York Times a few months ago by a colleague of mine, Peter Hotez, but this is the overall immunization rates in the United States by state for kids that are fully immunized by their second birthday. And so you can see that when we're looking at trying to get over this dark blue 75% or 80%, there ain't a whole lot of states that are doing very well on that level. And as a matter of fact, many, many states are actually looking at less than 2/3 of their kids are fully immunized in the United States. Now this all terrified us for a long time, but finally came to fruition, we thought, with a public health outbreak related to poor old Mickey and Disneyland out in 2014. And basically what happened is exactly what happens almost every day, although people don't realize it, is that some visitors came to a place that actually has relatively low immunization rates. So if you look at California, although it's a blue state that has large pockets of under-immunized populations, and very quickly as people then return from their wonderful vacations at Disneyland, then spread measles throughout the country quite quickly. This was thought to be sort of an unusual, newsworthy event, but it was something that we had actually known happens regularly throughout the United States. Although we declared measles eliminated in the United States in the year 2000, we actually know that travelers come in on a regular basis. This is the sort of year by year how many measles cases we actually get every year in the United States. Again, doesn't make a lot of press until it gets to the point where either there's enough of it that people care or when poor Mickey is affected. Now, measles is something that we're used as an example here because it is one of these things that we've done a very good job overall worldwide, where we used to have 2 million kids a year die. We're now down to about 250 to 300,000, but we still have large areas of the world that are actually under-immunized for measles. So teal, which is, I've been told, the dark green, when I had to ask somebody who knows colors, you can see that we're actually not particularly good compared to some other countries that actually have better measles immunization rates than we do. This is actually a worldwide problem, and as I mentioned, this is what I spend a lot of my time dealing with, is that 1 in 5 kids in the world does not have access to vaccines. For about $20, we could actually immunize each of those kids. Vaccines currently are preventing about 2 to 3 million deaths a year, but we still have about 22 million kids, unfortunately, in our country, including people that are then choosing to not avail themselves of these really valuable tools. Vaccination is one of the most important things we do to keep kids... Darn, I almost made it too, Laura. I'll at least get, whoop. So this is what I was going to end with. I was going to just try to get everybody, shots, shots, shots. Because this actually is what I think it sounds like usually when I open my mouth in front of my students, my patients, the people that I work with. Because as I mentioned, I'm a vaccine zealot. But there is really no better tool that we've ever developed to keep ourselves and our society healthy. And I ask for your endorsement and your support in trying to spread that message.
Jim. All right. Questions for Dr. Conway? I see some all around the room. I don't know where the microphone is. It's way in the back there. Okay, let's start lobbing it. There's some questions in the back of the room. We can maybe start there.
In terms of how likely you are to get infected, which is better, living in a rural community, an urban community, or a suburban community?
So it actually depends on the pathogen. So, many of the pathogens that we worry about for vaccine preventable disease are transmitted human to human. So basically, the more you're around people, the more likely you are to come into contact with people that may have those infections. So in general, rural communities where there's less human-to-human contact do tend to, in most circumstances, be less likely to acquire many of these things. However, there's plenty of these things that actually do get transmitted in the water and in food. So rotavirus, hepatitis A, doesn't matter where you are. And in fact, those actually sometimes are a little bit more common in some rural areas where the water supply may not be as well managed.
I will pass the mic along. Oh.
Bumble.
How do you approach patients or their parents that refuse to vaccinate?
Yeah, so we actually have, one of the things I was going to mention here. So we actually have a study that we worked on for quite some time here and actually have now implemented nationally. So one of the things that we realized was that it's unfair to people to threaten them. It's unfair to fire them and to dismiss their concerns. Because I don't really like the term anti-vaxxers. I think it's disrespectful. I mean, we ask people to be educated consumers, and so I like the term vaccine hesitant, where people have sort of done enough research that they've scared themselves, and so they benefit from information. And so what we've done is developed these refusal forms when people say they don't want to vaccinate their child that basically says exactly what that decision means, which is that I'm making a conscious decision to leave my child vulnerable to a disease that may disable or kill them, and that I'm accepting that responsibility. And when you put it in those terms and people understand and then can ask questions around that, we've actually decreased our refusal rates, which in Dane County are around 8 or 10%, to something around 3 to 4%, at least in the UW system where we've used that system. Again, we treat people with respect. We understand that they're doing the best thing. They love their kid just as much as we do, probably more. They're trying to make the best decision they can. It's just that they're not always as good at interpreting the information they get when they Googleize it. And so until that gives you a medical degree, we know better. It's just a matter of helping people interpret that information.
So when they're looking at herd immunity,
some people are going to respond appropriately to a vaccine, and then they get positive vaccine titers. So then some people are hyperactive in their response to that. And that can be related to people who tend towards autoimmunity. My daughter was an example. She became autistic 2 weeks after her vaccines. So some people do respond in a hyperactive way to lots of different stimulants, immune stimulants. Is there any research where they go in and see who responds differently to the vaccines, and then assessing that so that they might have, so those hyper people have a right to not vaccinate?
So first of all, my sympathies for your situation, and secondly, unfortunately, most of the research that we've done over now 40 to 50 years has really identified that autism and other neurodevelopmental issues that occur after people are immunized are really much more associations rather than causation. So if you have to respect the fact that in the pharmaceutical industry, where most of these products come from, vaccines are held to a completely different standard from other drugs. If I develop a lipid drug, I give it to 5,000 people, their lipids go down. I'm willing to tolerate if their liver enzymes get a little out of whack. But for vaccines, I'm giving them to completely healthy people with the goal of keeping them completely healthy. And so we don't tolerate side effects in vaccines at nearly the level that we do for other drugs. So when people ask why are vaccines so expensive, it's because they're tested on literally hundreds of thousands of people. And then we also have a post-licensure monitoring. So at any given time, 20 to 30 million kids are actually under active surveillance looking for side effects from vaccines. So these are kept at a completely different level than any other products that are used. The ultimate goal of vaccination, as everybody should recognize, is that we get to the point where we then no longer need to use those vaccines. So we've actually eradicated 3 diseases from the face of the earth by vaccinating and getting at least more than 90% of the population immunized. So we've eliminated smallpox, we've eliminated rinderpest, which is basically measles for animals, and we've actually now declared that polio type 2 has been fully eradicated. And so we've actually moved along in this by essentially effectively managing this and maintaining the population's well-being by balancing these risks.
Thank you so much, Jim, and that's
Thanks,
a great
Laura.
note to end on. Okay, so a few more notes. First of all, you all have been a phenomenal audience. Thank you. Give yourselves a big hand for acting out some of the concepts we were discussing. Before you take off, there's some more activities yet this evening. So as Patrick mentioned, in this room, he'll be talking about his book, Autobiography of a Disease. Otherwise, if you want to experience firsthand some more information and some more engagement with thinking about either bacteria or viruses, if you go out to your right, there will be the bacterial zone, and if you go to the left, there'll be the viral zone. I'm going to encourage our speakers to congregate in the zone that best aligns with their research, so that we can have the room again for Patrick's talk in just a few minutes. So we'll start that talk in about 5 minutes. If you want to stick around and talk with our presenters, Joe Handelsman and Laurel Legenza will be in the bacterial area, and Paul Alquist and Jim Conway will be in the virus area. Please join me in thanking again our speakers this evening.